Welcome back to my channel Bensil Biology. In today's video we are going to learn about mismatch repair. So let's get started with the lecture. During replication it can occur that DNA pol 3 can incorporate a wrong nucleotide that results into mismatch, and this needs to be repaired. This mismatch is first recognized by MUT-S, that is a homodimer who is scanning the genome. The complex MUT-S and mismatch containing DNA recruits MUT-L. Simultaneously, MUT-H recognizes and binds with GATC sequence that is present few base pair away from the mismatch site. As GATC is widely distributed along the entire genome, occurring once after every 256 base pair, MUT-H can differentiate between old and new strand because of adenosine residue of GATC sequence on parental strand is methylated, due to which parental strand is methylated and new strand is non-methylated. MUT-H is an endonuclease and is activated by MUT-L. The binding of MUT-H with MUT-L causes bending of DNA. MUT-H causes nick on new strand near the site of mismatch. Nicking is followed by recruitment of UVRD helicase at cut site of MUT-L. Helicase start unwinding the DNA, starting from the incision and moving in direction of site of mismatch. The separated single-stranded DNA is digested by exonucleases and stop just beyond mismatch nucleotide. Different nucleases are used depending upon where MUT-H has created nick. If it has created a cut at 5' site, then exonuclease 7 or Rec-J is incorporated, which degrade DNA in 5' to 3' direction. If it has cut at 3' site, then endonuclease 1 is incorporated, which degrade DNA in 3' to 5' direction. This section produces a single-stranded gap, which is then filled by DNA pol 3 and sealed by DNA ligase. The overall effect is to remove the nucleotide and replace it with the correct nucleotide. Thank you so much guys for watching this video. Don't forget to subscribe my channel and share this video with all your friends. Thank you so much.